We're seeing more and more news stories about all the different variants. We kind of lose track of them. There's so many now, and they keep talking about more and more different ones. There, first it started with the UK variant, the B.1.1.7, and then there was the South African variant, 501.V2, and then there's the Brazilian variant, Japanese variant. There seems like a new variant almost every day. And like I mentioned in the first video about the UK mutant, there have been like 4,000 variants sequenced. Most of them are of no clinical or physiologic significance, but these new ones, part of it is probably because we're sequencing more of them, and there is some press that can be gained by saying that there's another variant found, and it gives you some notoriety to the scientists that find them. But part of it is that some of these probably have some clinical significance, and this is a thing from NerveTag, which was published a few days ago. This is the group that first reported the UK variant. And when we look at all these variants, what we want to know is just a couple of things. One, we want to know, is it different somehow in its behavior? Like, is it more transmissible, and is it clinically more virulent? Like, does it cause more severe disease? Is it something we need to worry more about? And then two, does it evade immunity? Does it evade natural immunity, and does it evade vaccine-mediated immunity? Because that's very important to know. And so those are mainly the questions that we have. And at first, the original reports of the UK variant said that it may have less severe disease, but now reports are coming out that it may be slightly worse. This point 4 says there have been several independent analyses of these, and basically they report the hazard rate, relative hazard of death as 1.35. That means it's 35% more likely than the variant of concern. This one said that it was 1.29, and this one said it was 1.91, so twice as much. Now, this seems like a lot, but remember that the hazard of death with the case fatality ratio is about 3 to 4%, and the infection fatality ratio. So cases are basically known infections. We've tested them, and IFR, infection fatality rate, is basically patients that have infected. Many of them are asymptomatic. So if you include all of them, then the death rate is even lower, probably 0.5 or somewhat less percent. So a doubling of that hazard ratio isn't a huge jump in absolute numbers, of maybe half a percentage point to a couple of percentage points, but it does mean that they believe that it's a little bit more virulent. And a couple of things that we need to be careful in interpreting that is that the variant of concern was identified during a time when overall numbers were higher and the hospital and the health system was overloaded a little bit more. And we know that the death rates in California, which was also a little bit overrun recently, and fortunately the numbers are going down, so that's good, and the hospitals are not overrun anymore. But whenever a health system is overrun like that, the death rate will go higher just because of a bunch of factors. Patients don't get as good care when the hospital systems are stressed. So it's hard to say that the increased death rate was solely because of the biological behavior of the variants, or if because it just happened to, the variants came at a time when the health systems were overloaded. Because the death rate in California also went up, and that had nothing to do with the variants, most likely, because they were not a big part of the uprun. So these are two new papers. This is another paper out of South Africa looking at the South African variant. Now, this is a little bit misleading because it seems like they tested the actual variant itself, but when you look at the methods, basically, we'll scroll down to the methods. I think the methods are at the end here. Maybe it's at the beginning. Is it after the references? That'd be odd. Okay, yeah, here we go. So they put their methods at the very, very end. So what it shows is that rather than looking at the actual virus, what they did is they made a synthetic proteins with the same mutations as in the virus or the spike protein. So it's not exactly the same, but uh, it isn't the virus itself that they were testing. But anyways, what they found out is when they tested a protein that they had mutated to be similar to the virus protein, spike protein, that it was able to evade a lot of the known monoclonal antibody therapies that we have, and also evaded some of convalescent serum. So the important ones, like we talked about in a previous video, is this 484 mutation. Let's see right here, the E484K mutation. So that basically hides a lot of the epitopes, and epitopes are the parts of the protein that the antibody can stick to. And the K417 side chain was also seemed to be important, and that is also mutated in the South African variant. There is the other spike protein, the 501 position. That's the one that they hold the same as the UK variant. So it seems like the UK variant, like we talked about in the previous videos, is not going to be a problem in terms of immune invasion, but this seems like it is more likely to be a problem. And then this is another paper out of Rockefeller University where they did kind of the same thing, and they looked at the serum from vaccinated patients, so both with Moderna and Pfizer, and they basically looked at mutated spike proteins, and they found out that while the activity, so here we go, however, activity in SARS-CoV-2 encoding this E484K or the N501Y, so the mutations at the 484 and the 501 position. So the 501 position is the UK variant, and the South African variant has all three of these. Was reduced by a small but significant amount. So basically, the convalescent serum of these patients seemed to work, but not as well. So you needed more of this, a higher concentration of the serum for it to work. And they usually dilute these serums like 1,000-fold, and they still work. So that means we have plenty of cushion, according to this study. Now, again, this study did not look at the actual virus. And all of these studies are interesting because they give us some idea of what amino acids and what mutations may be more important, but it really doesn't tell us whether it is going to evade either natural or vaccine-mediated immunity, because this just tells us if the antibodies bind to it or not. And antibody binding is not the sole mechanism of action of our immune system. There's the whole cellular immune system with the T cells, and that may still work. And vaccine antibody binding may not be the only thing. So these are interesting studies, but we shouldn't say, oh, this study showed that the antibody still binds to it, so we're good, or this shows that the antibody didn't bind to it, so it's really, really bad. We won't know until basically the rubber meets the road, and that's when we start seeing, do people that have had the virus before, are they getting more infections with and when we sequence them, are they the new variant, or people that have been vaccinated, are they getting more are they getting COVID, and does it turn out to be the new variant? If we see a bunch of those, and we will see those coming down in the next month or two, that kind of information will become available to us. Then we'll know that yes, these things do evade our immunity. That would be a bad thing. And that's why the SIREN study that we discussed in a previous video is going to be really important, because they're prospectively going to follow these 20,000 people, check in with them every 2 weeks, and see if they are getting infected. So if their infection rate goes up and it turns out to be the new variants, we will know that these new variants are a problem. So I know we see a lot of these stories in the literature and in the news about these new variants, and there's a new variant like every other day, and I wouldn't get too excited about them, and I wouldn't get too excited about all these studies, which are scientifically interesting, but for practical purposes, not really changing what we're going to do, because we don't know what it does in terms of immunity, both from the vaccine and from previous infection, what it's going to do until we see them getting reinfected in people that have been vaccinated or have previously had the disease, and we will know that in the future. And the SIREN study, I think, is going to be one of the key studies, like we talked about. So I wouldn't put too much stock into it. Just kind of, it's interesting, but these papers don't tell us one way or the other for sure whether these variants are going to be a problem.